Professor Pumarola, what's new in clinical microbiology here at Medlab Europe?
Well, actually, at Medlab Europe, we are talking a lot about new technologies. I really think that this field is growing very fast in clinical microbiology, especially in the genomic field. And that's very important, and we are discussing that in this Medlab Europe, especially in two fields, how we are applying the genomics in emerging infection diseases, first of all, and in multi-drug resistant bacteria. And this is a very fast-growing field in the microbiology lab, and it's changing completely the horizon in the routine clinical microbiology.
Can you give us a few concrete examples with regard to new infections?
Well, for example, we have been discussing about Ebola, and we have been discussing about MERS coronavirus, etc., and how we have to do the surveillance using the new genomic tools.
So, can you describe how new genomic tools would be used, applied for these new diseases?
Well, this is not an easy question to answer, because we still have to increase our expertise in genomics. That's the first thing. The second thing is that we still have to make a good correlation between the phenotype. We know a lot of phenotype, but the correlation between pheno and genotype is not easy to decide. The cost. This is an increasing problem, especially nowadays. And we have to probably rethink and rebuild the whole specialty, and also the microbiologists. We will have to reinvent our specialty in order to apply the sophisticated genomics that are coming up in the lab.
So how will this change the routine, the work in the lab?
That will change everything in the lab. That will change everything. And we will say that probably not in the real near future, but in the midterm, we will see that how clinical microbiology will change completely because of the genomics.